So, hello everyone. I am Arnaud Pottersman from Instruct Center 1 in Strasbourg, in France. And so we are happy today to manage this session. So for those who don't know about Instruct, so Instruct is an infrastructure for structural biology, which provides access to researchers, but also training, internships, and funding for research and developments. So Instruct is a distributed research infrastructure with 16 member countries, so spread out all over Europe. There are in total 27 facilities. And so today we will have presentations from two users who access the Strasbourg Center. The first speaker is Katia Zanier, who will talk about mechanisms of IKK kinase substrate docking in the NF-kappa beta signaling. And then we will have a presentation by Constantin Brodolin from Montpellier University, who will discuss the regulation of transcription via RNA polymerase oligomerization. So with this, I think I will let the floor to Katia so that she can start.
Hello. So I cannot start the screen share while the other participant is sharing. So I need you are not to stop
I think I stopped sharing.
Let's see. Share. Now, do you see?
Yeah.
Okay. So good morning, everybody. So I would like, first of all, to thank the organizers of this web seminar for inviting me to present the results on kinase docking mechanism, which takes place at the very heart of NF-kappa B signaling. So this project has benefited a lot from Instruct infrastructure and Instruct support, financial support, and I would like to thank Instruct for all this. So I cannot change slide. I think I have to. Yes, here. Okay, so this is an oversimplified illustration of NF-kappa B signaling. So I first of all apologize for the horrible names of the proteins in this pathway. So I have tried to have a lot of Greek letters, so it can be quite confusing. You will find little schematic representations in many of the slides on the top right corner. So this should help you to navigate through the results. So what some of you might remember from universities textbooks is that for NF-kappa B signaling, there are two pathways, the canonical NF-kappa B pathway and the alternative NF-kappa B pathway. The inhibitor of kappa B kinase, also called IKK, which is the subject of this of our study, the focus of our studies, is a key regulator of both the canonical and alternative pathways. So this is a multi-protein kinase complex. In the canonical pathway, the IKK complex phosphorylates inhibitor of kappa B proteins, IKB proteins, which are the proteins that sequester NF-kappa B dimers. In the case of the canonical pathway, is P65, P50 in the cytoplasm in the absence of stimuli. So when the receptor gets stimulated by TNF-alpha, for example, IKB becomes phosphorylated by the IKK, and this leads to degradation of IKB proteins, leading to release of NF-kappa B diners, which in this case, have an exposed NLS signal, and then they are free to translocate in the nucleus where they activate gene expression. In the alternative pathway, IKK phosphorylates the precursor P100 protein. This leads to a partial degradation of P100 into the mature NF-κB P52 subunit. And again, there is an exposure of NLS nuclear localization signals, and with consequent nuclear translocation of the NF-κB dimers. So in addition to the functions in NF-κB signaling, IKK has many functions in other signaling pathways, and it's an important regulator of transcription, and for this reason is considered a pleiotropic kinase. So IKK complexes have a conserved catalytic core, which consists of homo or heterodimers of the highly homologous IKK alpha and IKK beta subunits. So in the canonical complex, we have an alpha beta heterodimer. In the alternative complex, we have an alpha homodimer. So we have to date structures of the homodimers of both of alpha and of beta homodimers. We don't have a structure for the heterodimer because of the difficulty in purifying this protein, this complex, which is has a high tendency to aggregate. So as you can see here, these dimers have a kinase domain, a ubiquitin-like domain, and then there is this large helical domain known as SDD for scaffold and dimerization domain, which is the one that mediates dimerization. So yes. Yeah. So despite the wealth of literature on this kinase, today there is no precise information published on the specific mechanisms of IKK kinase substrate interactions. And what we did when we started this project, we looked at the interactions between IKK and its best studied substrate, which is IKB alpha. And this substrate, this protein, has an N-terminal unfolded region which contains the phosphorylation motif, which is targeted by the beta subunit of the catalytic dimer in the canonical complex of by IKK beta, has an ankyrin repeat domain and a C-terminal unfolded region. So we have produced deletion mutants of this substrate and measured the interactions with by pull-down experiments with the purified homodimers of the beta, IKK beta subunit, which was produced with the help of the protein production platform at IGBMC by insect cell expression. So the results, so in fact, what we can see here that the interaction between, so these are homodimers which are nearly full length. What we can see is that the interaction with the full-length substrate protein is very transient. We have a very weak band in these pull-down experiments. However, if we truncate the first 73 amino acids containing of the substrate containing the phosphorylation motifs, we have an announcement of the interactions. We are not sure what this is due to yet, but anyway, this has been very important because from this construct, from this delta 73 constructs, we were able to further perform deletion. And in the end, what we came up is we could identify a small 17 amino acid peptide, which is found right at the C-terminus of the IKB alpha substrate, which is sufficient for interaction with the kinase beta homodimers. So this region contains a consensus, a conserved motif with a YDD phi-XV consensus. So we then wanted to see the impact to evaluate the impact of this conserved motif for the interactions between the full length or nearly full length proteins. And for this, we have performed in cellular protein-protein interaction analysis using the GPCA assay. This assay is based on the complementation of the split luciferase upon interaction between two binding partners. And so what we found is that, and this was not known at the time when we had these results, that both the beta subunit, which phosphorylates our substrate, but also the not only the beta subunit, but also the alpha subunit, which is present as in the canonical heterodimer here, is able to interact with the substrate. And the alpha subunit seems to be even have even a higher interaction. So again, if we delete the first 73 amino acids, just like in the pull down experiments, we have an enhancement of the interaction. And if we further delete, if we mutate, further delete the C-terminal region, or if we mutate the first and the third positions of the motif, so the tyrosine and the aspartic acid, we have in both cases, we have a stronger reduction of the interaction. So these data show that both IKK alpha and IKK beta homodimers recognize or dock to a conserved motif in the substrate, IKB alpha substrate. So we wanted to explore a little bit more this motif, and we created single amino acid substitutions at the conserved positions of the motif and measured the interaction with purified the homodimers by pull down. So what we see is that the alpha homodimers of the alpha subunit are more tolerant to substitutions within the motif consensus, and this is as compared to homodimers of the beta subunit. So this is somehow in line with the fact that the alpha subunit seems to interact a little bit stronger. But what we could also identify are two substitutions at the fourth position of the motif, which in the IKB alpha substrate is cysteine 308, which when they are replaced with a larger hydrophobic amino acid, like a phenylalanine or a leucine, provide an answer, make the interaction with the kinase dimer. And this was confirmed by ITC. So these experiments were in part performed at the biophysics platform of IGBMC in Strasbourg. This shows that this cysteine 308 leucine substitution within when introduced in the peptide containing the motif of IKB alpha leads to a reduction in the value of the KD value of about 24, both for the alpha, the interactions with the alpha and the beta homodimers. So, so this was very useful. We had identified at this point mutations in the motif that both increase the docking, the affinity of the docking interaction, and decrease. So in we have the C308L gain of function mutation, and we have this double mutation of the third and the first and the third positions of the motif, this YDSS, which basically suppresses the motif. So here, these are very good tools for functional analysis. And what you can see here is an in vitro kinase assay that uses endogenous canonical complex immunoprecipitated from HEC293 T cells. And what you can see is that the and it uses recombinant purified IKB alpha protein. So what you can see is that the IKB alpha substrate containing the gain of function mutation here in red is also displays the highest phosphorylation levels, while the YDSS suppressive mutation also leads to a decrease, a strong decrease in the substrate phosphorylation. So phosphorylation, I remind you, at the phosphomotif in the N-terminal region of I-kappa B alpha. So we went a bit further and we looked also what happened, what is the impact of the docking interaction on the degradation of the I-kappa B alpha substrate, which follows, in fact, phosphorylation of the substrate. And so these experiments were performed by our collaborator Laurent Chariot in Liège and used mouse embryonic fibroblasts that are knockout for the I-kappa B family genes. So in this, we have reintroduced I-kappa B alpha proteins in this mass by viral, so by retrotransduction. So what you can also see here, that the degradation of I-kappa B alpha is slowed down when there is this YDSS suppressive mutation. So what we conclude from these studies is that the docking affinity correlates with both phosphorylation of the substrate and degradation of the substrate. So this motif has a strictly conserved tyrosine at the first position. And so we wondered why this is the case. And we found an early report that shows that this tyrosine, which is tyrosine 305 in the I-kappa B alpha substrate, is phosphorylated, and the phosphorylation is enhanced upon activation of the C-Abl kinase, which is by genotoxic stress. So as you can see here, so we measure the impact of tyrosine phosphorylation by ITC, and what you can see here is that when we have a phosphopeptide with the first tyrosine of I-kappa B alpha with this first tyrosine of the motif phosphorylated, we have a strong decrease of the interaction with IKK, in this case is IKK alpha subunit homodimers. So it seems that in fact this tyrosine phosphorylation can act as a negative regulator of the docking interaction. So we were also able to confirm phosphorylation of this tyrosine in the I-kappa B alpha substrate at steady state. However, we were not able to determine what happens upon stimulation of NF-kappa B canonical signaling by TNF alpha because of a limitation of the assay we use, which requires pervanadate treatment. Pervanadate is a phosphotyrosine inhibitor, and this also it happens that the treatment by pervanadate also inhibits NF-kappa B signaling. So next, we wanted to see if peptides containing the YDDFXC motif can inhibit NF-kappa B signaling. And so we constructed these fusions where we have I-kappa B alpha peptide containing the motif fused to a E3 sequence, which forces dimerization, homodimerization. So we created in this way a bivalent peptide, which is able to interact stronger due to avidity with IKK dimers due to avidity effects. And so we transfected these fusions. So we also put M-scarlet for detection by immunofluorescence. We transfected this fusion as mRNAs in fibroblasts, and we looked at nuclear translocation of P65, which as a marker of activation of NF-kappa B signaling. So of canonical NF-kappa B signaling. So as you can see here, we observe P65 nuclear translocation only in the presence of I-kappa B alpha peptide with the suppressive YDSS mutation. So without our negative control peptide, in fact, without the motif. However, a peptide cysteine 308L mutation, so this is the gain of function mutation, blocks P65 in the cytoplasm, just like in the unstimulated conditions here, where we did not add TNF alpha. So this shows that these peptides are able to block NF-kappa B signaling, but also tells us the docking is active during signaling. So somehow, coming back to this tyrosine phosphorylation issue story, this tyrosine has to be non-phosphorylated during activation of NF-kappa B signal. So it's indirect evidence. So to summarize, we have identified a docking interaction mediated by motif. And from the early studies, it seems that this docking interaction is negatively regulated by pathways, genotoxic stress pathways that impinge on IKK complexes through phosphorylation of the motif. So we have also performed structural characterization of the interaction between IKK dimers and motif peptides. So this we have performed many attempts of crystallizing this complex. This was done with the help of the Instruct biocrystallography platform at IGBMC. So we were able to raise crystals in many cases, but the crystals were not diffracting well. We were able to obtain just one data set at medium resolution, which allowed us to reconstruct the dimer structure. However, due to this poor resolution, we were not able to see clearly the peptide. We were only in the case of one of the homodimers in the asymmetric unit, the AB homodimer, we were able to see a little extra electrodensity lobe, but in the middle of the SDD domains of the dimer. But we could not do much about this with this. So we used another approach to determine the binding pocket of the motif on the dimer. So for this, we collaborated with the Yuri Rapsilber in Berlin. We performed cross-linking mass spectrometry using 3 different cross-linkers with different spacer arms and chemistries. From this, we derived about 30 unique distance restraints, and which map, in fact, all map to the SDD domains of, so these are restraints between, of course, the peptide and the IKK dimer. So these restraints all map to the SSD domains of the dimer. And since, so this region, they map, many of the restraints map to this very region, actually, where we saw this extra lobe of electron density. We performed, so in the X-ray structure, we perform data-driven ad hoc modeling using the crystal structure of the AB homodimer that we had solved, plus the cross-linking mass spec-derived restraints. So the modeling was done by ad hoc, and what we can see is that the motif sits in a groove which is contributed by the 2 opposing SDD domains of the IKK dimer, IKK beta dimer. So we explored a bit more this interaction by mutagenesis of the kinase, in this case of the beta homodimer, of the IKK beta homodimer of the kinase. So what is the most important, the mutants, the IKK beta mutants were tested for interaction with the substrate, I-kappa B alpha substrate, so either in cellular using our truncated delta 73 kappa B alpha construct, or in vitro with purified proteins using a peptide of I-kappa B alpha comprising the docking motif. And probably the most striking result is this double mutation, L654D, W655D. This is a mutation that was shown to abolish, to suppress IKK beta dimerization. So this mutation also suppresses binding to the substrate in both assays here, as you can see here. So what we can conclude is that dimerization is required for substrate docking and is consistent with our finding of structural finding of the docking groove being contributed by the two SDD domains. And we could, in addition, could also identify some other positions that are in the pocket that are important for the interaction. So the motif is, we found that it's not only present in the IKB alpha substrate, but also in the IKB beta paralog, which is also a substrate of the canonical IKK complex in canonical NF-kappa-B. We also detected the motif in P100. P100 is the substrate of IKK in the alternative NF-kappa-B pathway. And in addition, in IRF7, which acts in the interferon pathway, and this has been shown to be also a substrate of IKK alpha. So we confirmed these interactions by pull down, as you can see here with purified proteins. The P100 and IRF7 peptides interact with IKK alpha homodimers, but not with the beta homodimers. This is coherent with the biology that the functional data that show that they are substrates of IKK alpha. We measure these interactions by ITC. We find a very weak interaction with the IRF7 peptide above 100 micromolar. However, the interaction between the P100 peptide and IKK alpha homodimers is in the low micromolar range as in the same range of value of the other interactions with the IKB substrates. So we also further studied the contribution of the motif in P100. So what the motif is present in the N-terminus region of P100. And if, again, if we introduce mutations, we introduce a YDSS suppressive mutation within this motif within the P100 full-length protein, we can see here from the in cellular protein-protein interaction analysis that we have quite a strong suppression of the interaction with the IKK alpha subunits. So to conclude, we have identified a novel docking motif with a YDD-FXV consensus, which mediates docking, which is found, in fact, specifically in NF-kappa-B substrates of the IKK kinase. So we don't detect it except from IRF7 in the other substrates of the kinase, and this is interesting because for drug design, because it could be a way to specifically target the NF-kappa-B functions of IKK. So this motif mediates the interaction with the catalytic core dimers of the IKK kinase, seems to have higher affinity for the alpha subunit, and this also explains why the alpha subunit is always present in the IKK complexes. Can be negatively regulated by phosphorylation at the first position. And we were also able to show that bivalent peptides derived from this motif can inhibit canonical NF-kappa-B signaling. We also have provided quite solid evidence that this motif binds to a groove at the interface of the IKK dimer. thus also providing a function for dimerization, IKK dimerization in NF-kappa B signaling. So I would like to thank the people who have contributed to this study. So Changqing and Stefano, who have done are the principal authors, and also the other people from our lab, our collaborators at the University of Liège, at the Institute of Cancer Research in London, and at the Technical University of Berlin. And here, all the people, the engineers and the scientists of the Instruct platforms in the Strasbourg area. And I thank you for your attention.
So thank you, Katja, for this nice presentation. So there are a few questions. Do you see them, or I should read?
So. Maybe better that
Yeah,
you...
maybe I read them, or?
Yes, you read them, because
Yeah,
I lost you. I think I have just to expand.
So the first question is there evidence that IKK subunits are cleaved and act as independent domains in cell?
No, they can't, because the domains, in fact, they are required. So there are a lot. If we look at the structure of these dimers, there are a lot of interactions between these domains, and these are very important for the function of the kinase. So if we, for example, if we just isolate the kinase domain of IKK, it will have a much more, it will target different phosphomotifs. So it will have a much more, less specificity, let's say. So because it seems that they are partially unfolded, they require the other domain. There is an intricate network between SDD domain, kinase domain, and this ubiquitin-like domain.
Okay. So there was another question: was did you try AlphaFold to model these multimers or the binding of the peptide?
Yeah, so we tried. It doesn't get it. It puts it on. It puts the peptide on the kinase domain mainly, because you have docking also can. There are many other instances of docking. I mean, structurally not so many, but we know that docking motifs dock to grooves within also the kinase domain. So this is but in other cases it goes through separate domains present in the kinase, so like SH2 domains, for example, or other subunits like with cyclins.
We have a few other questions. We'll go quickly. Does the substrate binding of the affect the stability of the dimer when you bind the?
This probably yes. I think it should it should close it a bit, so make it a bit, because all this problem with diffraction seems to arise from the opening, and there is movement. This dimer has a tendency to be, and we see in the structures in the asymmetric units, there is one dimer which is more closed than the others. So this breathing of the dimer is a problem for diffraction. And yes, so one of the things, it's possible substrate binding that should close it or make it more stable.
Okay. So with this, I think we'll finish this session. There was another question, somebody who raised the hands, but I don't know how to.
Yes, they are allowed to talk now.
So can Aegun, can you speak? No. Okay.
So there is a comment from Philippe Dumas.
Yes, can you?
Now I see. Okay.
So Philippe says that the ITC with the phosphorylated peptides shows a delta H, which is almost 0. He says, which does not mean that the interaction is lost, or on the contrary, it may be a maximum affinity.
Oh, okay. I have to see with Philippe this. Okay, we can discuss.
Maybe we can discuss in private.
It's just downstairs.
And just Ken Agun, I mean, she wanted to speak, but okay, no. And Pierre Poussin, a last person, Pierre Poussin, raise his hand, but no.
They are allowed to talk. They will just have to unmute.
Okay, or maybe it's maybe they don't want. It's an error, maybe. Anyway, so with this, I think we can thank Katja again for this presentation, and then we can switch to Konstantin.
Okay.
So thank you, Katja. Very much.
Thank you. Bye-bye.
Bye.
Can you hear me?
Yeah, we do hear you.
Okay.
But
so wait, just need to start and to share my screen. Okay.
yes.
Okay. So I can go, yeah?
Go ahead. Yeah.
Okay. So hello everybody. I'm Konstantin Brodolin. So I'm working in Institute of Infectious Diseases in Montpellier, and so I'm going to present you our recent work on transcription on Mycobacterium tuberculosis. So first, I would like to thank the organizers for giving me this opportunity to present our studies. And first, I just want to remind you that tuberculosis is one of the global health problems. A lot of people are infected over the worldwide, and the mortality of the disease is high, around 1.5 million per year. And in particular, the main problem now is there is an increase in the number of multi-drug resistant cases of tuberculosis, which basically can't be treated by any of known antibiotics. So there is an urgent need to develop new drugs targeting this bacteria.
Just Konstantin,
yes.
We don't see you. I mean,
you don't see me. Okay. Sorry. Yeah. Okay.
Thank you.
So, so basically, one of the ways how Mycobacterium resist to antibiotics is based on its tricky transcriptional regulation, and which is really now it's popularly understood. And so we are working on basic mechanism of transcription in order to understand how we can actually develop new drugs targeting tuberculosis. So as you know, the transcription is the first step in gene expression, and it is performed by RNA polymerase, which is a huge molecular machine. So basically, RNA polymerase is composed of the catalytic core enzyme of 5 subunits. So where you have largest subunits like beta prime, which form active site cleft holding the DNA template. And also these subunits also form a channel for RNA where nascent RNA came out of the active site. But to initiate transcription on double-stranded DNA, and to recognize promoters specifically, RNA polymerase requires additional factor, which in bacteria called sigma factor, which binds to core and form whole enzyme. So in mycobacteria, the specificity is that the first step of whole enzyme assembly, it's also controlled by the additional transcriptional factor called RBPA, which basically binds to core and binds to sigma also and stabilizes whole enzyme. And this factor is essential for bacterial growth, which basically it's an essential factor, and it is involved also in resistance of bacteria to stress and to antibiotics. Paradoxically, in fact, there is also a second transcription factor, which is called CARD, which also bind to RNA polymerase, and also essential for bacterial growth and implicated in stress and antibiotic resistance, and also formation of the persistent sleeping bacteria. When these two factors bind to whole enzyme, this whole enzyme can efficiently recognize promoter DNA sequences, which in bacteria composed. So there are two motifs, -10 and -35 motif on promoter, which are recognized by the sigma subunit region 2 and 4. And to initiate transcription, RNA polymerase must open promoter DNA, so to create a single-stranded DNA in order to initiate RNA synthesis. And RBPA and CARD are implicated in this process. They are basically essential for RNA polymerase to open promoter DNA. But mechanics of all these steps is really still poorly understood. And more complex than in mycobacteria, there is in fact certain sigma factors of different structure. So there are 4 groups of sigmas, but here we will focus on only 2 sigma factors, which are sigma A, which is basically housekeeping sigma as principal factor, which basically control transcription of or more, at least 70% of genes in bacteria. And also the second factor is sigma B, which is stress response sigma factor, which is induced by stress, and also it is used by RNA polymerase during exponential growth phase. And but paradoxically again, these 2 sigmas are almost identical. So their main promoter binding regions 2 and 4 are identical and can recognize the same promoter motifs on DNA. The only difference for sigma B is the shorter N-terminal region 1.1. So the question is why we need two sigmas, in fact, and is there a difference in the specific regulation between the sigmas? Because basically both sigmas also are combined to RBPA and are controlled by RBPA and also CARD. So what makes sigma B regulation different from sigma A? And this was actually, so our studies is focused on sigma B because it's a really poorly studied factor. And we suspect that it can be implicated, play an important role in formation of antibiotic-resistant bacteria. Because there were a lot of studies on sigma A, in fact, in particular structural studies, but almost no on sigma B RNA polymerase. So we decided to go structural analysis of sigma B RNA polymerase. So, but first we performed just negative stain electronic microscopy on our samples of RNA polymerase core and RNA polymerase whole enzymes. So here you can see that in solution, RNA polymerase core and sigma A whole enzyme are presented as monomers. So you can see perfectly well this RNA polymerases. But surprisingly, when we look at the sigma B whole enzyme, we observe that it forms large oligomers, which are flower-like. And more interesting is that when we added RBPA to this mixture, we see that oligomers dissociate. So there is something happening with the whole enzyme here. And so just the question was, what are these Palau-like oligomers? So we decided to go to cryo-EM. And so we, I forgot to say that we, in fact, we use recombinant RNA polymerase. So we constructed an expression vector with all subunits of RNA polymerase core. So we can express it in E. coli and reconstruct the whole enzyme from this with sigma subunit. After, so we went to Titan, we got access to Titan cryos in Strasbourg at the IGBMC. And so basically collected images, and after to declassification cryospark, you can see that we have nice flowers of RNA polymerase oligomers here. So next we went to ab initio reconstruction of the three-dimensional model, and here we got nicely this structure, which actually was an octamer. So we have 8 RNA polymerase units which are actually stacked. So there are 2 rings of each of 4 units which are stacked together. So when we perform the refinement in cryospark using the symmetry, so we got a nice map of the RNA polymerase octamer at 4.4 angstrom resolution, so which is quite good for this huge complex, because you can imagine that we have 8 units, so it means it's a complex around 3.2 megadaltons. So here we see this map colored according to local resolution, and what it says that in fact that central part of the map is well resolved, so it shows high resolution between around 3 and 4 angstroms. However, extremities of the RNA polymerase units are really poorly resolved. So we have resolution up to 8 angstrom and higher. So it means that there is a high conformational mobility in this complex, and this actually doesn't allow us to resolve the major part of core RNA polymerase. Also, another problem here that we have, as you see, that there is an angular distribution of particle projections of this, and you see that there is strong preferential orientations in this mixture. But however, it was okay, because I mean it was not really limiting in this case, because we have a symmetry. So concerning the conformational heterogeneity, in fact, we decided to see what happened with this complex. So we performed analysis of heterogeneity analysis in cryospark, basically by using a subset of the particles. And so here we can do to see how the conformation of protein changes in 3 dimensions over these particles. And in fact, what we observed that, so here you can see the 2 conformations of the octamer in different dimensions. And in fact, we see that the protamers are moving relative to each other in this octamer. And this actually, it was a major problem that we can't get high resolution structure. So to bypass this problem, we performed a local refinement. So in fact, we took initial model and initial set of particles. We performed refinement of this model without symmetry. So we got this low resolution map around 6 angstrom. And in this map, in fact, the protamers, there are two protamers which were better resolved than others. So we focused on these protamers. So we created the mask on the one protamer and on the dimer, so we can better resolve the interaction between these RNA polymerase units. And then we performed refinement on this masked structures, perform local refinement, and this allows us to get nice structures, high resolution structures. For example, for RNA polymerase monomer, we have 3.8 angstrom structure. And basically, we can see in this structure, so it's shown here, we can see that most of the core now shows resolution between 3 and 4 angstrom, which is quite good. And basically, this from this map, we managed to create the model of RNA polymerase whole enzyme, which is shown here with all subunits. So here we have beta prime subunit, which is also the major binding site for sigma. But for sigma B, in fact, we see only part of the protein. Basically, it's region 1 to 2, which interacts with the -10 promoter region. But we don't see the C-terminal part of the subunit, suggesting that this part is flexible, is moving around, and it doesn't fit to the RNA polymerase core. However, yeah, after so this we using this model, we reconstructed the whole structure of the octamer, so we can see all the interactions between RNA polymerase units in this huge structure. And what was interestingly, in fact, if we have a look on these interactions in details, so here's an example of the interaction between the protamers R1 and R5 in the octamer. So we can see that the sigma subunit of R1 protamer interacts with the core elements, core RNA polymerase from R5 protamer. And in particular, what we observed that beta flap domain of RNA polymerase core interact with promoter binding region 2 of sigma, while beta prime zinc binding domain of RNA polymerase core interact with non-conserved region shown here in yellow. And this region, in fact, is the site for RBPA binding. So here, for example, if we compare this with a model of RNA polymerase promoter complex and RBPA, so you can see that RBPA occupies the place of zinc finger domain, and beta flap, in fact, interacts exactly in the place where promoter DNA should bind to sigma. So basically, it means that these interactions should block binding of RNA polymerase to promoter and inhibit its function. So to get some functional insight on this an effect of octamer on promoter binding and transcription, so we perform some mutagenesis in the NCR domain. In fact, what we observe that if we compare the housekeeping sigma A with the sigma B, so in the NCR domain of sigma A, there is insertion, which is which is shown here by it's encircled here, and this we think that this insertion, because we compare the structures, so we see that this insertion should block octamer formation. So we introduce this insertion in sigma B, and we have a look on this ability of sigma B mutant to form octamers, and in fact we observe that insertion indeed prevents octamer formation. So basically octamer can be formed. So this is perfect agreement with the fact that sigma A doesn't form octamers. So next to check if there is effect on the activity, we use a stopped flow fluorescent assay. So basically, you take promoter DNA fragment, which contains a size 3 dye incorporated in the near the -10 region. So in the region which should be melted by RNA polymerase. So basically, when you form open promoter complex with a single-stranded region of DNA, the fluorescence of size 3 will increase. So next, so here you can see the kinetics of open promoter complex formation. So we can see that for wild-type RNA polymerase in presence of RBPA, we see a strong increase in fluorescence, as well as for sigma B mutant in presence of RBPA. However, wild-type sigma B, wild-type enzyme alone cannot form open complex, as you can see here, while mutant sigma perfectly well form open complex even without RBPA. So there is still activation effect of RBPA even with a mutant, because in fact RBPA has a double function. So it can, first it acts on the sigma subunit. So it basically allows insertion of assembly of sigma to RNA polymerase. But second, RBPA directly stabilize interaction of RNA polymerase and promoter DNA. So basically, this perfectly well. This results shows that in fact there is an effect of oligomerization on RNA polymerase activity. So finally, so this based on these results, we propose a model suggesting that in fact sigma B acts as an inhibitor, in fact, of RNA polymerase activity. So basically, it promotes spontaneous oligomerization of RNA polymerase core into inactive state, which maybe it's some kind of a storage of RNA polymerase in the cell. Only when you have a binding of RBPA to this structure, it will lead to dissociation of the whole enzyme of this octamer and lead to the formation of the active transcriptionally active whole enzyme, which and finally lead to the burst in transcription in the cell. But still, yeah, we are working now on getting the proof in vivo to support this model. Anyway, yes, I think this is the first example of the formation of this these huge structures of RNA polymerase, because there were several reports on the dimerization of RNA polymerases and also ribosomes, which is used for regulation of gene expression, while there were no cases on formation of the higher order structures like this. So, so finally, I would like to thank the people of my team who were involved and this contributed to this project, Zaki Marishol, Oran Shaluan, Rishi Vishwakarma, and also collaborators from CBS, and also I'm grateful to access to Instruct for giving me access to IGBMC platform in Strasbourg, and so thank you for your attention. Yeah, that's it.
So thank you, Konstantin, for this very nice presentation. So maybe I can start by a question. So sigma A is larger than sigma B, if I get.
Yes.
And so did you, and if you truncate sigma A, do you get the same? Do you also tend to?
Yeah, this is a very good question, in fact. Yeah, this is one of the hypotheses to test. We are working on it now. So still, yeah, for the moment, I don't know, but we have some problems with expressing truncated form of sigma A. So as soon as we will solve this problem,
Okay,
yeah, but
thank you.
yeah, I can expect that maybe it can be like this.
And so there is a question by Patrick. He asked if the oligomerization mechanism is found in other bacteria.
Yeah, well, I can't exclude it, in fact, yeah, because in fact here the regions of RNA polymerase which are involved in oligomerization are basically universal. They're highly conserved between all bacteria. So I can expect that maybe another bacteria can exist. At least I believe that in actinobacteria, because Mycobacterium is belong to actinobacteria, and they all have RBPA factor. I think that for all actinobacteria it can be true. Yeah.
But for other bacteria, do they also have a long version or a sigma A-like and sigma B-like?
Actually, yeah, actually, almost all bacteria have two sigmas, so they have a sigma A-like and sigma B-like. Yeah, and we still, yeah, it's so actually a big question why all they need two sigmas, because yeah, it's really, yeah.
And just physiologically, how is the expression of sigma A and sigma B controlled? I mean, this is because you mentioned it could be a storage mechanism. So
Yeah. Yeah, sigma A, in fact, is yeah, it's housekeeping. So basically, sigma A is also controlled by itself, yeah, and also by sigma B. But sigma B expression is controlled by other sigma, which is the stress response. So it's a really complex network, which and is really poorly studied. But what we observe, in fact, that both sigmas, because before it was there was a notion that we think that sigma B was only stress response or stationary phase. But now the last studies, in fact, show that they both co-expressed at similar levels throughout the cell growth. So it's so basically they can coexist at the same time. And yeah, the question is why? Yeah.
okay. So with this, I think we can. There are no more questions that I see on the chat. So maybe we can close this session and thank again Katya and
Can you hear
Konstantin.
me?
Oh yeah. Now we can hear you.
I apologize for the previous mess. I'm the same person who was trying to ask a question, but my microphone didn't work. So for this one, I would like to ask, well, I would like to comment also. It's a very interesting phenomenon that you have seemed to discover, but I'm just confused as to how one should interpret it. Usually, the enzymes that I am familiar with, I'm a PhD candidate, by the way, I'm a rookie. They tend to have a sort of bilateral symmetry, and that actually completes an active site usually, doesn't it? It makes the enzyme functional instead of dysfunctional. In this case, does the enzyme inhibit itself? Like, is it sort of an aggregation? Like, what's the interpretation, the right interpretation to have of this phenomenon? It binds to the transcription factor, then it binds around that, like, morph itself, creates some radial structure, and that structure is somehow inhibited. Like, that's the point of my confusion. I would appreciate some clarification. I really don't understand how to interpret the phenomenon. Like, what category is this?
So to understand right, so the question is Is it this? So this structure is actually, I think I didn't mean. Okay, so can you repeat?
Okay, I'll try to simplify. Maybe I have no point at all. I don't know.
Yeah.
I'm seeking some clarification here. So the transcription factor bound monomer is functional, right?
Yes. Yeah.
And only when more of the enzyme binds to the complex does the system get inactive in that hibernation state.
And actually, yeah. Well, let's say so you have a sigma binds to core and creates a whole enzyme. So it shows there is a low activity without RBPA. And basically, it spontaneously goes to the octamer. So
Right.
and after this, it's really basically it's not active at all. Yeah. So you need
It's just nothing I've ever seen before.
Huh?
It's interesting. It's not nothing I've ever seen before.
Yeah. It's,
Thank you very much.
yeah. Okay. Thank you. Yeah.
Okay. So if there are no more questions, then we can close the session for today. And thank again, Konstantin and Katya. And so see you in February or for the next Instruct meeting online.